To computer. Okay, so hi, Gurdeep. Hello.
Hi, Lisa, how are you?
I am good. How are you?
Doing good, doing good. Yeah, excited for this event and excited to see you after a long time. Yeah, so it's good. Yeah.
Yeah. So we met as part of our PhD programs, respective PhD programs at the University of Toronto. And
yeah.
part of the Connaught and Vanier Scholars community. Yeah, and since then, you've continued the academic journey. And maybe you can share something about that. And we're talking.
Sure.
We're talking about 20. It was 2015 or 2016, something like that, when we met.
Oh, actually 2014-ish. Yeah, I think so. I think we were in the same graduate house when we moved in, like starting the PhD. So I moved from Boston, and yeah, I think you moved from UK, I guess. So yeah, I started PhD in cell and system biology department, and we both had Connaught scholarship, and then you went ahead and got a Vanier scholarship as well, which was very awesome and very prestigious. So that's great. So I finished my PhD last year in 2021, and after that I moved to Vancouver. I'm working at BC Cancer as postdoctoral fellow, and my work is on cancer epigenetics, decoding the epigenetic basis of drug-resistant cancer and how the cancer pathogenesis start, what is the epigenetic basis of that, or molecular drivers of cancer in the epigenetics. So yeah, so my PhD work was on epigenomics and genomics analysis of stem cells and other tissues. We're identifying enhancers which control the fate of those cell types, which are enhancers. So these enhancers control which gene will be expressed in which tissue. So its code is very difficult to identify. It has not been explored much deeply before, but when I came to the field, I used machine learning and comparative epigenomics and other tools to decode the enhancers, and I created some synthetic enhancers which work as equally as natural enhancers. So natural enhancers has capacity like this, and the enhancers, synthetic enhancers which I made, also have capacity like this. Previous synthetic enhancer has capacity like very low. So now we can say that we know the code of enhancers because we can make synthetic ones also, and we can understand the natural enhancer pattern also based on my model. So after that, I wanted to apply all this expertise and knowledge in enhancers and epigenomics to the epigenetics to a real life problem, which is cancer. So I'm excited to do that here at BC Cancer. Yeah.
Now, my question is, how do you explain everything you do to someone who knows nothing about epigenetics?
Okay, so epigenetic is like you have a big kitchen with a lot of spices and all the ingredients, but when you're cooking some specific dish like sweet dish, then you won't use like paprika spicer in it or some other kind of things which will not make it sweet anymore. So that's the same way we have 25,000 genes in our like genome, and each cell has same 25,000 genes, but only few thousand, like 5,000 or something, which are expressed in high amount, which make these proteins, which make that cell's identity. The same way, like when you're cooking, you don't use all the ingredients in your kitchen, but you specific only for that specific dish or whatever cuisine you're going to make. Same way our cells also make those specific cell types with the help of cell-specific proteins. And those proteins are when to make those proteins. So that is controlled by these enhancers and promoters, which act as a switch. And those switch are controlled by these epigenetic marks, which could be either DNA methylation or histone modification. So when they could have either active state or repressed state. So if it is in repressed state, the epigenetic is in repressed state, then the gene will not make protein. So transcription will not happen, so the protein will not be made. So that so your whole body as a if somebody is cooking it. So for that, you need a precise control of what thing to take, what kind of spices to put and what not to put. And that is all controlled by the epigenetics, which epigenetics has two states, like active or repressed, or it could have the third state also, which is like poised state, which with little flick, it will turn to active state very fast. So those help in transitioning from one thing to other thing, or one cell type to other cell type. So the stem cells in your body, when the zygote is formed, that make all kind of cell of your body, including heart, brain, liver, eyes, like everything is made from those from stem cells. But it's the same genetic code, but it's different cook with different specialty comes, and then so those are the transcription factors, different proteins which make those specific cell types, help in directing stem cells to different specific differentiated tissues like heart, liver, or brain, or pancreas, or all those kind of things. So yeah, that's where epigenetics, not only their epigenetics is important in making different cell types, but also is important in daily life also. Even in memory formation, there are some epigenetic or epigenomic changes which happen in your hippocampus brain for a like in 20-30 minutes period, which reverts back to its naive state in like 6 hours-ish. But that changes what happened, kind of makes the memory in the brain. So all this switch on and off are happening all the time in our body, and the epigenetics kind of controls that. Yeah.
It makes me think about language, and at least about how generative linguistics treats language, right? We have this parameters that are either switched on or off, and based on that, we are going to be able to
exactly
a language.
So yeah, exactly, this is very true for genomics. Like, so these enhancers you can think as some sentences, and the whole genome is like a book. So there's a famous quote from Eric Lander that bought the book hard to read. So once the genome was sequenced, like now we understand what is it saying, what is its function, what does it mean. So to understand that, it's same thing like linguistics. Like, but here the problem is that we don't exactly know what is the grammar, what is the vocabulary. So what kind of vocabulary, how the sentences are made, how the essays are made. So that's what we're trying to decode, and where these enhancers act as one of the sentences, which is made of multiple words. And those words are like transcription factor binding sites where the protein bind. So there's a specific grammar, there's specific vocabulary which you need to make a functional enhancer. So when I was making synthetic enhancer, I was making sure that like I'm using specific vocabulary of transcription factor binding sites, which are coming together in specific manner, that they have some kind of grammar in them or not. So all those kind of things I was taking into account, and it worked. So yeah, it is very close to the linguistics, you can say that. So it's the language of a genome. Yeah.
How old is the field of epigenetics? Oh, how you
that's a good, yeah. So I would say like in last 60-70 years, like everything has accelerated from like, since we identified the DNA structure. So all those things have been happening in last 100 years, like what is DNA, what is nucleus, but slowly the structure of DNA was identified in 1950s, and then after that sequenced the DNA for the first time, and then the first enhancer was identified in 1973 or something, or 1981. So yeah, so since then, like from 1980s, the field was started growing. Previously, the enhancers were more studied in the B cells or immune cells, because the switching is very important for immunity. But after that, we identify that they are important not for only those cells, but many other cell types. So other cell types, tissue-specific enhancers were identified. And then slowly the whole genome sequencing kind of very helped a lot. So in 2001, the whole genome started sequencing. So we had the mouse genome sequenced and human genome sequenced, and RNA sequencing done. So from the 2000s, it started exponentially growing, because now we have this sequencing tool which you can use along with the experiments and answer many questions. So one of that method is ChIP-seq, which was more explored in 2009, where you can use this technique like in any cell type to with the help of sequencing to identify where a transcription factor is binding. And with the help of that, you can identify where these enhancers or those regions where epigenetic changes are happening or epigenomic changes are happening, where histone modifications are getting repressed or active. You can identify that based on sequencing. And now there are some new version of that technique, which were recently made, like cut and tag, which was published in 2019. So now that method requires very small number of cells. You don't need a big tissue. Even with a few, even with 1,000 cells, you can identify what is the epigenetic state of that cell type, because sometimes there's tissue availability issues, especially from the patients and stuff. So the field is advancing, and we are using newer tools, like transposase that is used in cut and tag. It's getting better and better and more complex parameters you can sequence now at the same time. Previously, only one kind of modification you can sequence at one time, but now you can do 2 or 3, 4 together. And you can also now sequence a single cell status. So previously it used to be in bulk. So you have thousands of cells combined. You are like just sequencing all of that together. You don't know if they are just one type of cells or there are 2, 3 types of cells mixed together. But now you can do that even in single cell. What is the epigenomic state or epigenetic state? And then you can identify how many types of cells were there and how different they are and what different functions they were doing based on that. So the field is advancing with every year. It's getting bigger and bigger. Yeah.
How is it?
So
It's interesting because it's very different, for instance, from a field that is fairly established, right? This field of epigenetics is really it's growing by the hour, and you're part of the field. You're part of this growth. So I wonder, I would like to hear your experience of being part of this growing field. How do you experience this?
Yeah, I'm very excited to be part of this growing field. My PhD was on it kind of in epigenomics area, and my postdoc is on epigenetics and epigenomics, both combined, DNA methylation, histone modification, all those combined with enhancer, with the actual physical implications like cancer and drug resistance to the cancer treatment upon chemotherapy. So there's no limit. There's like you can do multiple things because the applications are very diverse. Not only just one tissue, it's multiple tissues, all kind of diseases. Sometimes have the roots in epigenetics. Yeah, it's very exciting to be in the field right now and learn. Sometimes it gets like you have many techniques, what technique to use, but once you get mastery in one, then you can apply that to multiple cell types. And also it's easy to learn the new techniques because you have already mastered the previous version. So, but if you start from scratch, it's the learning curve is a bit more like difficult. But yeah, if you have mastered previous techniques, then it's easy to switch over to the new technique. So I think that's the only thing you have to look into, but mostly otherwise it's very exciting because the more tools, the more answers you can get, more deeper you can go into these questions or rabbit holes. Yeah.
And the applications that you're talking about, at least from what I've understood, is the drug resistance of the illness, right? And how so maybe you can say more about it. How does the field of epigenetics and epigenomics helps us to become better at dealing with cancer?
Yeah. So what happens in the cancer treatment, like when you use chemotherapy, then there are some cells which don't die, and they don't die because drug resistant. They're drug resistant because they're different kind of epigenetic makeup. So their different epigenetic makeup make them a bit different, which make the drug does not work on them. Now, because those are still cancerous cells and they have different epigenetic makeup, they will keep on growing in the body, and then they will spread all over the body and they'll kill the person. So now, until we decode what is the epigenetic difference in the drug resistant and drug sensitive cells, we cannot actually use drugs more effectively. We have to use drugs in combination with the drug against the normal cancer, which will kill 90% or 99% cells, but that 1% which will again grow and spread to the whole body. For that, you need some to target some of the epigenetic regulators, which target the differential epigenetic landscape between the sensitive and resistant clones. So once we have those drugs which target those transcription factors or regulators which regulate the epigenetic landscape specifically in the resistant ones, then we can kill both kind of cancerous cells. So that's the main goal. And also it's diseases. There are many other diseases also which are like the result of epigenetic changes, which happens lots of neurological diseases, and there's some mutations which happens which also cause these diseases because they lead to the different epigenetic changes. For example, one mutation in an enhancer which control the limbs, so that can make you have like multiple fingers. But if that whole enhancer is deleted, then it is done in like mice experiment was done in mice. If you delete that one enhancer, the mouse will have no limbs. So it will, and that enhancer is naturally deleted in snakes. So snakes don't have any limbs because they have that enhancer deleted during the evolution. But if that enhancer is deleted in the mice in the lab using genomic methods, then that will have no limbs also. So it kind of shows like how just even one enhancer is so powerful. But think about, we have thousands and thousands of enhancer, like so maybe 100,000-ish for all different cell types combined. So the even one base pair mutation can have drastic effects. So the potential is a lot of there, but also like how during injury, during injury, also epigenetic changes happen. For example, you have an eye injury, but that changes something in your eyes, then you can't see anymore because the cells have become adapted to a different epigenetic state. Now they can't do that original function anymore. And other examples could be the inflammation or immunoinflammation and COVID, long COVID. I hope some more tests are done on the epigenetic side of it, how the epigenetic changes are happening in different organs, which make that long COVID process. So many diseases which happen actually have roots in the epigenetics. When we shift from the right path, the cells shift from the right path, then when they're diverged and they are in that state, they cannot do the normal function anymore. So it has all the other disease phenotypes then, yeah.
So it sounds like science fiction, actually.
Cool. Yeah, but yeah, it's all happening in our body all the time. But yeah, we're just trying to like decode it now slowly. We're identifying the function of all of these things. So yeah, like one enhancer which was deleted in mice and the chimps and humans. So that made the difference, like why the brains of chimps and humans grew bigger as compared to mice. So that was due because of one enhancer deletion. So some of things like happen randomly sometimes, but all of these random changes or deletion of insertion of new enhancer, they bring a lot of phenotype effects. So all these species which we see, they don't have much changes in the protein coding regions, but it's the changes, changes in the enhancers which make them different. The speciation events happen mostly due to the evolution of enhancers rather than the protein coding regions. So yeah, so we're just trying to explore that right now. Yeah.
You said, you spoke about earlier about how to actually produce a drug that would be effective on the cancer cells that are drug resistant. We will have to really decode the epigenetic changes, that the mutations have taken place in these genes, and then develop a specific target drug that would be different from the previous the drug that was affecting other cells. And I wonder how are you working in terms of so you have the epigenetics lab. Are you collaborating with a different lab from pharmaceuticals, like and then doctors? How does it work?
Yeah. So in my current lab, in Dr. Simon Aparicio lab, we have a great reservoir of the PDX samples. So PDX are like patient-derived xenografts, where the cancer tissues are taken from the patients and they are engrafted in the mice. So the cancer tissue, human tissue, grows in the mice, and then it is taken from the mice, and then experiments are done on that. So, and our lab also have like many other tools for like doing the epigenetic analysis, like for RNA-seq, we can do RNA sequencing, but we send the samples to the sequencing center. So all the rest pre-processing is done in our lab for RNA-seq or ATAC-seq or cut and tag, which I'm doing right now, which to identify the histone modification, epigenetic changes. And our lab also do how the copy number changes based on sequencing. So what kind of mutations are happening in individual cells. So all that is done in our lab. Like for the sequencing of the DNA, we have this new technique called DLP+, which our lab employs. So we do most of the processing in our lab. Then when we have the library is made, then all we need to be done is sequence. So there are different sequencing facilities. So for example, for BC Cancer, we have Genome Science Center. They have a big sequencing facility. So we just send them sample, and in 1 or 2 weeks, they give the sequencing results back, or even faster if the UBC also has a sequencing facility, VRC. So we send that. Sometimes they give the sequencing files in like 2 days or 3 days, so they're fast. So all the steps are done pre-steps, pre-processing, and like processing of everything, making library is done in the labs. So there are different kits and different chemicals you have to order, but there are some equipments like 10x machine, which we have, so which helps in the single cell resolution of these epigenomic profiles. So we have that. But we also collaborate with the other labs also. For example, like New York Genome Center, they're making this new method for cut and tag, where you can identify the landscape of multiple histone modification at the same time in the single cell. So I got some of the reagent from them. So I'll be applying that on the cancer tissues which I have, and then send it for sequencing and then analyze that. So after that, you have to do a lot of computational analysis to decode what is meaningful, because once you get the results, there is sometimes a lot of noise. So you have to do a good right statistical analysis, machine learning, all of those kind of steps have to be done to get to the signal. And that signal is the actual, and then you have to validate that signal that it is working. So you can knock down that protein or increase its expression. So you can do those two methods, loss of function or gain of function, to prove yourself and everyone that this target which you identified as signal through all that process of sequencing and everything, machine learning, that it is functional. Once it is, you prove it 2-3 times, like at least 3 times to prove it that this works, then you publish it. And the next step is, there are some drugs, if you know that they are targeting that same target which you identified, then you can use those drugs. You can try 5 or 6 different types of drugs which you think will work on this pathway or something. Then when you try and test that, then you can see that, okay, this drug is killing most of the drug-resistant cells. So then it goes to the next step, yeah, for for the testing in the patients and other things, or clinical, yeah.
Yeah. So were there cases in which you actually that you were able to help a patient due to the epigenetic drugs that you've manufactured?
Yeah, not myself, but the lab where I'm working now, they have this one drug which was published in a paper in Nature Communication in 2016. So there's this drug which is now in clinical trials and is effective. So, and yeah, so hopefully that will be available for the clinical, for the actual patients soon after the trials are done. But yeah, I'm new to the field, like where the drugs are used for all this stuff. But previously, my work was on more genomics, epigenomics, understanding the enhancers, which I did in the stem cells and other tissues. But now that is done, now I want to apply that to the cancer and drug area. So hopefully we will find some good therapeutics which we can apply for drug-resistant cancer. So that's the main goal. Yeah.
I'm asking specifically because my dad is now coping with drug-resistant cancer for the past 2 years. He has liver cancer, and chemo doesn't seem to work. So
yeah. So yeah, it's a big, huge problem. Like once this holy grail is resolved, then I think all the cancers could be beneficial, because right now mostly the cisplatin, the platinum-based chemotherapy is used, but mostly the drug-resistant cancer evolves somehow. So we need to target that, and once we target that, yeah, I think we can save a lot of lives. So I hope to be part of that process. Yeah.
Mm-hmm. I'll be happy to hear about the name of the drug, at least, kind of to.
Okay, sure. It's a little bit different name, but I will tell you one sec.
Sure. Maybe others will be able to benefit from it as well.
Yeah, no, definitely. Here. Yeah, it's called CX5461.
Can you type it in the chat maybe?
Yeah. CX-5461. So yeah, so this is a DNAG quadruplex stabilizer. Mm-hmm. So it helps in the patients with the BRCA1 and 2 deficient tumors. So yeah. So that our lab recently published in 2017, and it has promising results. Mm-hmm. Yeah, it will be exciting to see how things go.
It doesn't have yet the name, like the market name, right? So it's still I'm guessing
it's like a chemical compound name.
Yeah. So it's still fairly early in the clinical trials, I suppose.
Yeah. That's true. Yeah.
Okay. It's very interesting. It's very for me. I'm curious to hear about what led you to this field to start with. How did you end up in this field? Yeah.
Yeah. So I was always fascinated by the biology, like cell biology and DNA when I was in high school, and then I did my engineering in biotechnology, where I was doing a lot of courses and laboratories where working with like molecular biology and other kind of fields like immunology and plant sciences. So doing all that, trying different projects. I did like multiple projects during my undergrad to identify what fields excite me the most, and then I went to like US Boston during after the end of my third year. So I was working with viral genomics, and I was fascinated by genomics. Feel like there's so much potential to sequence virus and all those kind of things, using molecular biology to decode what is the function. So I was very fascinated by the DNA sequences and genomics whole field. And after exploring the viral sequences, then I thought, let's go to the bigger DNA, which is exploring the function of human genome or mammalian genome, mouse genome. So that's why I joined the lab of Dr. Jennifer Mitchell at University of Toronto. So there I was identifying the code of enhancers in stem cells and other tissues. Yeah, I tried multiple methods. So in the end, using different unique methods and unique approach, I was able to decode that, and I'm happy that it worked. So yeah, so this publication which we did in Genome Research, yeah, in that I decoded the code of enhancers in stem cells. So yeah, it was a great journey. So yeah, enjoyed it. Yeah. So after that, yeah, I knew like, yeah, this is what I wanted. Genomics, epigenomics. That's what I want to do. Epigenetics. That's my passion. And yeah, I'm still continuing that.
So
yeah,
it's evident that you're passionate about it, and it's really exciting to meet people who are passionate about what they do and research and their work. And I'm curious to hear, when was the first time that you heard about biology, that you learned about biology as a young person, you know, that made really light up and say, okay, this is where I'm going?
Yeah. I think when I was a kid, I used to watch a lot of Discovery Channel, which used to have a lot of biology shows and everything. So like a lot of like DNA related, cell biology related. So when the first time I was reading biology, like a textbook about cell division, mitosis, meiosis, like, yeah, this is very cool. This is very fascinating. The chromosomes, genes, and all that. So yeah, I was very fascinated by that. Like, we need to know more how the life works. What is the sequence code of life? Like, we are made from that DNA, but we don't know how it all works. So yeah, so that led me to like, we need to explore DNA more. And so yeah, that's why I went with the viral genomics, small genome first. Like, viral genomes are smaller compared to the human genomes, but yeah, I'm happy to have going up the ladders and getting more complex and more complex. Yeah, so yeah, it's fun.
So 2 questions about that specific story. One is, how old were you? And 2 is, where in the world geographically was it?
Like when yeah, I was in India like that time. I would be like maybe like 7 to 10 year old, like, since I've been watching all that, like it made me fascinated, like, yeah, I should learn it more, but I didn't know what to do it with it, how to do it. But like, this is fascinating. You should do more. Like, there is more. Like, yeah, this is what I want to do, kind of thing. I didn't know exactly what to do. And then went to do undergrad. Like, did a lot of course, a lot of labs, like a lot of chemical labs and chemical courses and things. So I was trying to explore like what I want to do, what thing exactly in biology I wanted to do. And then went with molecular biology. It's like, this is interesting. This is fun. This is like more mathematical. There's like yeah, it's more logical. So, and all those things which excited me more. And then I started going to the sequencing. I was like, this is much more cooler now. So, and using computational tools, bioinformatics, computational biology, coding, and yeah, that excites me also. The coding, I love coding. So I do most of my coding myself, machine learning myself, which also makes me help understanding things more deeper. Because if there's some people who just do the coding work in biology, like they don't know much about biology. And there's some people who are doing biology, but they don't know how to analyze that thing, or what is the interpretation of that whole sequencing. So they give that work to this, the other people give that work to this, and I try to do both. So I think doing both helps in understanding the depth of each, and I can make customized coding for whatever thoughts I have on biological aspect. And it's hard to give that thought to someone else. In the time I'm interpreting that problem to someone else, what they will do will be completely different and not do that depth because they don't have that depth of the biological knowledge. So yeah, I think that is a good strength, I would say, that I can do both, like wet bench work, all the experiments myself, and do the computational coding and machine learning, all those myself, which helps me go deeper in both sides, where both things merge. I think that's when best thing happen, when multiple things merge together. So yeah.
It's almost like I'm thinking in linguistic terms, because this is kind of something that is more familiar to me, even though I did my in high school I took biology classes, and I took in Israel there are like you can expand on a subject, and the subject that I studied more in depth was biology, and specifically genetics. My mom used to help me with the studies, because it was so hard for me to understand. And my mom was she was a pediatrician, and for her it was easy.
Definitely.
But I'm light years away from biology in my understanding, and I'm thinking about language and how you speak about decoding the genome, and I'm thinking that there are different layers of interpretation, different layers of stories that are that can be harvested through the work that you're doing. We spoke about machine learning, right? Was that
machine learning.
It's coming from the computer sciences, right?
It's a computer science like new field where now it's like you can use AI and other things, but there are many other tools which are developed by statistics and mathematics and computer science, where machine learning like lasso, random forest, all those classifiers which are used to identify what features are important. So that field, I think that has revolutionized kind of many things. But biology specifically too. Like biology uses all the new methods which are created in computer science. They're all kind of they land up in biology to because they have to be applied to some problems. And what deeper problem could be the life or sequence code, which has evolved over hundreds of millions, 100 million years, So think that how many different paths these sequences might have taken in 400 million years to get to where we are, and we're trying to decode that in our small life period. So it has to be done with high computational speed and other things.
It's really impressive how multi-dimensional, multidisciplinary your research is.
And definitely, we can't do all that. I think if sequencing capacity was not there, if previously people used to, if you have to sequence things, people might run things on gel even for small base pair, like 1,000 base pair, but sequencing. But now we have these big machines like Hi-C, and they're like Nextera, all these big machines. Like now they can sequence GBs of data in like few hours. It's like 10 to the power 9 versus 10 to the power 3 in like few hours. And so there's a great mechanical side to it also. This mechanical engineering combined with chemical engineering, with optical engineering, all that combined help making this machine which can sequence the human genome like 2 hours-ish, like those. And then there's like nanopore technology where you just need with a small thing, you can sequence your whole genome like in one single strand in few hours. So yeah, all that technology is helping to giving all that data what we are trying to explore the function. So yeah, so yeah, I think we need still there's lots of things to do. Like the more we know what we don't know, and when we start exploring what we don't know, we open new doors. And then we know this part, but again, there's 90% of this unknown. Then you try to identify that unknown. So I think this process keeps on going, but yeah, a lot of progress is being made, and I think implications for health is coming there, which is good. So yeah, it's exciting right now.
I'm thinking from a perspective of like maybe it's philosophical, maybe it's this literary criticism aspect that the world I'm coming from, and it sounds to me that just from what you were saying, that as you said, there are different layers, and if you interpret it, if you work with the genome, you come up with one sequence, you come with one story, as it were.
Yeah,
and
exactly. Yeah.
if someone else was to do this, they would have a different story to tell.
Yeah. No, that's a very important topic you touched. So yeah, so what happens, like, especially not with even like with different people, like if you're using different method, for example, the answers which I will get from RNA sequencing will be a little bit different than what I will get from DNA methylation, will be different from what I will get from the histone modification of one type, and then there are multiple types of histone modification. So all the answers which I get from different things will be a bit different, because all of them bring some of the noise with them, because they are not the perfect phenomenon of the biology. These are the methods to reach that central target, but they come with own of their noise. So in the review article from Jay Shinder lab, he mentioned this problem as blind men inspecting elephant. So if there's an elephant and there are 5-6 blind men are inspecting it, someone will hear like take its trunk and then think, oh, what is it? This is like is it like a snake or python? Someone will take its tail. Is it a broom? Someone will touch its like leg. Like, is it a trunk tree? So everyone will have different kind of thing, but because everyone is kind of using is partially blind, but when you see as a holistic picture, where everyone is coming through, then you might realize that, oh, this is an elephant. So all these methods, like, yeah, so that's why I think computational coding is also very important, because, and statistics, with that you can try to identify what is actually meaningful non-random thing, which is away from the noise. And once you remove the noise, and that same thing is being validated by multiple areas, like the guy who's touching the tail is also finding that thing that there's something big or something huge. It's an animal, it walks or something. So if all of them are coming to the same conclusion, that's where the real truth is. The rest is the perception of what you think is truth is. So there are so many false positives and false negatives and false positives, and so all those things are there.
I was almost waiting for us to get to this question of truth. Because yet again, from the world, the discipline I'm coming from, right? There is no truth. Right? In literary analysis, there is no truth. There is a bunch of interpretations. Reality, and each person has their own interpretation. It's somewhat impacted by philosophy, by psychology, but by all kinds of humanities related fields. And then comes science, that is supposed to be this capital T truth. And then the story you're sharing with me tells me, here too there are these false positives and false negatives. There's this this narratives, this multiple narratives that come together, and we need to figure out which ones are actually hold holistically.
Yeah, absolutely. Yeah, like I mentioned you that technique like ChIP-seq, which people use to identify enhancers. So if you use ChIP-seq of transcription factors, you'll find that only 20% on average would be functional. So it has true positives are only 20%, the rest are false positives. So they are not positive. If you try to test them, they will not give any result. But which are true positives? They will be functional every time you do it. So in a sense, I think the truth would be what is replicable. If any person, if I'm doing this assay, you're doing this assay, and anybody else doing this assay under the same conditions, like hopefully if you could describe those same conditions, same kind of chemical environment and everything, then that thing should give same result. So that is truth. But these methods on global level, if you only one method on global level usually have a lot of false positives. So, and again, so one thing you can say is it's a noise. And second thing would be like it's a reminiscent of the truth in some other cell type. So maybe that 80% of what we're seeing is not functional. Maybe it's functional in some other cell types, but it still have some of the, it's not, the signal did not fully disappear in that specific cell type. So there's some crosstalk, but it's kind of nice. So, but somebody may say like, yeah, this is functional because it's functional in some other tissue. So those are the things where where sometimes the definition could be contradictory. Yeah.
Very interesting. Very, and again, it takes me to this world of language and how we interpret what we see around us, and how everything in my mind translates into this linguistic sequencing, into this grammar that if we understand the grammar rules, we can actually understand the parts of speech that construct the sentences, the structures that are around us.
Our DNA is a book of stories, and some stories make our childhood like how our body will behave like that, how our bodies will behave when it is adult, and how it will behave under some conditions. And during old age and all those things.
So we have 10 minutes or so left.
Sure.
And I want to be selfish, but I'm sure it will benefit others as well. I personally am worried about having Alzheimer's, simply because my mom had an early onset, and my grandma didn't have an early onset, but she had Alzheimer's too. So if we think about epigenetics, what are the probabilities, considering that you know nothing about my genome, but from your professional point of view?
For Alzheimer, like there are two things. So like one thing I would say is the gene therapy, not gene therapy, but the precision medicine is one thing. So you can take these cells from your skin, you can convert them to stem cells, and those stem cells can be converted to neurons. And that, when they're neurons, it could be injected in the brain. So this is being done by Blue Rock Therapeutics company, which was started by some professors of University of Toronto. So we're working with stem cells and differentiation of the stem cells. So they're trying to make some neurons and heart cells. So if you make some neurons, you can inject them in the brain. So those kind of things, hopefully, I think they will be in trials soon, and that can definitely benefit a lot. And the other part is the epigenetics side. So there, I think there are, some studies being done. There's like professor in MIT, Professor Manolis Kallis. I think he has published some recent papers on Alzheimer based on single cell studies. So, and he took out published the epigenome landscapes of the Alzheimer and the non-Alzheimer models. So I think all those things, once they are taken to application level, for example, now we know the epigenetic landscape, but now if that could be used to identify some drugs which can make the epigenetic landscape of normal versus the Alzheimer one back to the naive state. So I think there's potential. So now there's more studies happening in this area. So I think there should be some epigenetic drugs, or even if some genes are identified, we can knock those genes down using CRISPR methods, and all those things are those tools I think will be, hopefully they will be used for therapeutic purposes. I'm hopeful that therapeutic field will be able to utilize that soon. So there are a lot of good things are happening, and especially you can check Manolus Callis, some recent papers. So he has done a lot of work on Alzheimer's. Recent papers at least.
Well,
sorry,
could you write his name as well?
Yeah, I will write that.
Thank you. I will definitely check it out. Okay, thank you. I'm also thinking about it sounds like epigenetics, much like many other fields in biology, are targeted towards therapeutic goals, right? And I wonder, once we heal cancer, once we heal Alzheimer's, which are right now, I'm not speaking about COVID because this is something that's being resolved as we're speaking. This is how it sounds like or looks like. But Alzheimer's and cancer and HIV and the illnesses that have been this great peril for many people, and great source of fear. What do you expect that is going to happen once these are healed? What is going to surface? Because something needs to happen, right? Something needs to.
I think aging would be the next target. I think there's some epigenetic plays some role in the aging as well. So I think that will be next target, like how we stay young forever. I think those kind of things, or how we can reverse aging, those kind of things might be, they might make expensive drugs for that, but if it is a functional working, I think that will be a cool thing too. So you can predict how old that person is, maybe based on epigenetics, those kind of things. Some could be done. You can identify the brain's epigenetic status with the help of the epigenetic results that you get from the blood. So there are many things you can do with cell-free DNA in that. So there's a lot of potential for all the things like what your body is doing with the help of blood. Can you identify that or not? So there was this one paper which especially showed that there's correlation between the brain's epigenetic and the blood's epigenetic. With the help of your blood, you can know the brain's epigenetic status. So there's a lot of potential, especially in the age field. That's what I can think of. But there are many diseases, maybe others which need to be conquered after that. So,
and I mean, there is so much that might happen that we don't even know about, right? Some surprises that could resurface.
I think as many organs we have, I think that's as many potentials we have for epigenetics, and because it is used in all the organs, everything. it's kind of the secret code which keeps things in the right order. Like even when you go to sleep and everything, there are some transcription factors which work in circadian rhythm, and if that is messed up, that can mess up the other rhythms and things. So yeah, there are a lot of potentials. I think we just need to just start looking into it, and there will be potential there. So yeah, it's just like we're not looking there yet. So yeah.
Fascinating. Thank you so much, Gurdeep. I really appreciate your time and your passion and your sharing. Yeah.
Yeah, my pleasure. It was great to talk to you, and yeah, great discussion. And I love the analogies are there between genome and linguistics. Yeah, the grammar, like the sentence vocabulary, all that things come up because genome is like a code book. So we're just trying to decode that book, and we're not there yet, but we are getting closer day by day. Yeah.
Great.
Cool. Thanks.
So
Yeah.
much.
Yeah. Thanks. Take care. Yeah, great pleasure. See you. Yeah.
Bye. Thank
Bye.
you.